Welcome to SS Educare Teaching Learning channel. Thank you for all my subscribers. Those who are watching without subscribing, please subscribe and watch my videos. Your support is my strength. Let's go to our channel. Story of the day. There was a king with one leg and one eye, but he was very generous and liked by all the people around him. He was a tough competitor for all the rulers. One day, when he was walking through the hallway, he saw the portrait of his ancestors. He also had an intention of, what if I have my own portrait in front of mine? I'll be very happy to see me every day in such a big portrait. So he called all famous painters across his kingdom and told the same thing. Those painters were confused and to say that the king had a one leg and one eye, and how come they will draw such a beautiful portrait? They all refused and went. But a young artist came forward and said he will bring out a beautiful portrait of the king. The king gave him 3 days time. After 3 days, he came and unveiled the portrait in front of the king. The king saw it and was very happy, and he didn't expect such a beautiful portrait of himself with so many abnormalities in himself. What could be the painting? What could be the painting? Think and tell me. Yes, the creativity of the painter or the artist was awesome. He actually made the king to sit on a horse with one leg focused and one eye closed, aiming at something. That is, he was having a bow in his hand and an arrow ready to shoot. So he is aiming at the arrow with one eye, and the other eye was closed. So he has drawn in such a way that his one leg and one eye was completely not shown. See the creativity of the painter. The king was very happy and he gave him so many prizes. So the moral of the story is, you have to look at the positive aspects of anybody, ignoring the faults or drawbacks of them. If you look at positive aspects, everything would be positive around yourself. That's the story of the day. In my today's video, I will teach part 3 of chapter 2, cell cycle, cell division and structure of chromosomes. The first 2 is been over in my first 2 videos. The third part is structure of chromosomes. Coming to the word chromosomes, the word chromosomes means chromes and soma. The word chromosomes are divided into 2 parts, chromes and soma. Chromes meaning color. Soma means body, which means when you stain these chromosomes with dye, the whole body of chromosomes will be colored, will take up the color and observe the color and will be seen with colored for us. That is why chromosomes were given this name. So this name chromosomes was coined by Waldeyer in the year 1888. As he saw or observed the chromosomes the first time under microscope, he coined these 2 words. Next, this chromosomes contains chromatin and histone proteins and the DNA. Chromosomes are nothing but chromatin with histone proteins and the DNA in it. This chromosomes was first discovered by, you may ask me, what is this Waldeyer? Waldeyer is the one who coined the name as chromosomes. But the chromosomes, this is what is chromosomes. Chromosomes are nothing but the condensed form of chromatin, was told first or discovered by Voltaire Fleming in the year 1888 itself. So, this chromosomes was as the result of cell division, that is chromatin fibers during cell division condensed to form chromosomes. See, look at the structure of chromosomes. I have drawn arms with green color and the center one, the arms are joined at the center with, I have drawn it with red color. So, these two arms of chromosomes are called as chromatid, joined at a center called centromere. Inside the chromatids, it has a genetic material or the fluid part called as chromatin. Next is structure of chromosomes. First, in the structure of chromosomes, you have chromatid. We already discussed chromatid is arms of chromosomes. The two arms, one in the upper end and another in the lower end, is your chromatids. Next, chromonema, the bead-like accumulation. See, if you see, the bead-like chromatin material are called as chromonema. Next, centromere. Centromere, as the name says, it is in the center, joining the two arms of chromosomes. See, look at this. This is your centromere. Next, chromatin. The fluid material present in the arms of chromosomes are called as chromatin. This chromatin is divided into two, euchromatin and heterochromatin. Euchromatin means when we are staining it, the portion of chromosomes which stain very lightly. In this picture, if you can see, these are the lightly stained portions. They are called as euchromatin. And these are the darkly stained portions. This darkly stained portions are called as heterochromatin. Next is size and shape of chromosomes. The size of chromosomes ranges from 0.25 micrometer, this symbol is micrometer, to 30 micrometers. The largest, this 30 micrometer, the largest chromosomes are lamb brush chromosomes. In fruit fly, the size of chromosome is 3 micrometers. And in man, it is 5 micrometers. So for us, the size of chromosomes is 5 micrometers. Then in maize, it is 8 to 12 micrometer. Next, position of centromere. Based on the position of centromere, chromosomes are divided into 4 types. First one, telocentric chromosome. In telocentric chromosome, the shape of chromosomes are rod-shaped. Rod, that is from top to bottom, it is of same size and same shape. Then, the centromere is located at proximal end. Proximal, proximity means at the tip, topmost, at its extreme end. Look at this, this is the centromere and it is the proximal part, the topmost part. Down you have your chromatids. This is a telocentric, you call them as telocenter. Next, acrocentric chromosomes. Acrocentric chromosomes are also rod-shaped chromosomes and the centromere is at one end. If you see, centromere is being sent through one end and the other arm is very small and the second arm is little bit longer. So it is B, acrocentric chromosome. The third one is submetacentric chromosome. It is T-shaped or L-shaped chromosome. Look at the shape. It may be T. This arm may be here and forms a T shape, or this arm may be completely, if you look, it is L-shaped. So the centromere is at middle. Not exactly the half or exactly at the center, but it is at the middle where it can be of T shape or L shape. That is your C, submetacentric chromosomes. Next, your metacentric chromosomes. As the name says, it is exactly at the center where the two arms are located, one at the top and another one at the bottom. So like this. So this here it is bent like this, but if you see the two arms are equal. They are metacentric chromosomes. They are V-shaped, bent. During cell division, we have seen all the chromosomes lie in an equatorial plane in this shape. Metacentric chromosomes in this shape, V-shaped. Centromere forms two arms. That's your fourth type, metacentric chromosomes. The next is karyotype and ideogram. These two are sister words. Karyotype means the group, it is a type of identification of chromosomes based on few group of characteristic features. Okay, they are identified as a particular set, that's called as karyotype. Karyotypic diagram consists of chromosomes like this. If you look at this diagram, this is one chromosome, this is one set of chromosome, and this is one set of chromosomes. And here if you see, these chromosomes are very, very small. And this chromosomes, if you see, both the arms are equal in size, little bit larger than the first one. The third set of chromosomes, the upper arm is short and the lower arm is longer. So based on this characteristic features, that is the arms of chromosomes, they are characterized or they are divided into A, B and C. So this type of categorization is called as karyotype. Next is ideogram. Ideogram, this is also a type of identifying chromosomes based on their morphological characteristics. What do you mean by morphology? Morphology means the structural identification. If you see, same whatever I told there, here if you see, position of centromere can be one thing, or the type of dye it is taking up. Assume here the chromosome has taken a blue color dye. All chromosomes do not take up blue color dye. So maybe based on the dye it has taken, based on the position of centromere, based on the size of the arms, whatever it is. So based on the morphological features, we are classifying or identifying the particular set of chromosomes. So that type of identification is called as ideogram. Next, chromosome number. We divide chromosomes or we call chromosomes into two categories. Number one is haploid, where you have only N, which means initially I have drawn centromere with only one arm in the upper end and one arm in the lower end. That is called as a haploid. Before cell division, they will be in haploid condition. After cell division, maybe during the cell division, the chromosomes are getting duplicated. The same single arms of chromosomes are getting duplicated, one above and one below, and then forms diploid, that is 2N condition. The N chromosomes are duplicating into 2N. So therefore chromosomes are called as haploid, N condition, and diploid, 2N condition. Next, chromosome in human beings. In human beings, it is different, identical when compared with others. In human beings, we have 23 pairs of chromosomes. Pair in the sense 2. A pair has 2 chromosomes. 23 pairs means 46 chromosomes we have in us normally. In that 23 pairs, 22 pairs are called as autosomes. And one pair is called as allosome or sex chromosomes. As the name says, this one pair or one set of chromosomes determines the sex for the gender for the human beings. Whether we are male or female is determined only from this one set of allosomes or sex chromosomes. Male chromosome, if you see, it has X and Y pair, and in female it is X and X chromosomes. Based on this, we categorize into male and female for the human beings. Next is importance of chromosomes. Chromosomes are important for, number one, they form a link between two generations. They form a link, that is, chromosomes have genes. The genes are transferred from one generation to another, from grandfather to father and from father to their son. So there are similar genes, very few similar genes are transferred from one generation to another generation, so that the generations are being linked by chromosomes. Next, second one, these chromosomes would induce variations through crossing over. I already said in my previous video what is crossing over. Crossing over is nothing but the non-sister chromatids, they cross during cell division. They bring about variations. Example, I already said, example is if I take a hibiscus plant which has an ability to yield more number of flowers, and in the other hand I have rose plant which have an ability to give different colors of flowers. When I cross these two different characteristic plants, I will get a hybrid plant, a variant where it has the characteristic feature of giving more number of flowers and different colors of flowers. That is what is variations. So these variations are definitely possible and beneficial for the commercial people. So these chromosomes help in bringing out variations through crossing over. Third point, they control the synthesis of proteins. Which is the one that helps in the synthesis of proteins? It's our ribosomes. So these chromosomes have a check, an eye on the synthesis of proteins also. Proteins are not being synthesized over the limit. Next, these chromosomes would suddenly undergo mutations. Mutations are nothing but the sudden changes occurring to the structure of chromosomes. That brings out evolution of new life. Definitely, if there is some change in the chromosomes, there will be the evolution of new organism, different from the other one, the parent one. So that brings out new species actually. From that new species, you may get new other species. So that's the evolution of new life. So these chromosomes would undergo mutations, the sudden changes, maybe addition of some gene or deletion of some gene or the repetition of some gene. All that is possible because of mutations. So that would lead to, that contributes to evolution of life, in the sense evolution of new variants, new organisms. The next important topic of this chapter is structure of DNA. Before talking about DNA, let's talk about genes. What are genes? It's the unit of heredity. Let me talk about heredity. What is heredity? Suppose in the first generation, grandfather. Next in the second generation, we have father. In the third generation, we have grandson. Some of the grandfather's features will be there in grandson, who is in third generation. How is this possible? It is possible because some of the genes what grandfather is having will also be there in grandson. So how is that grandfather's genes will be there in grandson? Passing of these characters from grandfather to father and from father to son. That is transformation of these. How is this possible? This is possible by units called as genes, which this transformation or transfer of this characteristic features from one generation to other generation is called as heredity. Who is helpful for this process of heredity? It's the genes. That's why it's called as unit of heredity. What does these genes have? It is composed of DNA. You call it as deoxyribonucleic acid. Deoxyribonucleic acid is nothing but your DNA. There are almost 30,000 genes in human beings. Then this structure of DNA, so whatever I have drawn here, this structure of DNA was first proposed by Watson and Crick. They told that the DNA strand is wrapped around histones. I already said in my previous video, the strands of DNA will have a proteins or histones in it. So when DNA wrapped with histones, we call that whole unit as nucleosome. That is, DNA with histones are called as nucleosomes. Then the two strands, DNA in its structure, it has two strands. Number one, this is your first strand, and second one, this is your second strand. The first strand is starting from 5' direction and ending up with 3' direction. So it ends with 3' direction. The second strand starts in the opposite direction, that is your 3', and it ends up with 5' direction. So you have two strands of DNA which is coiled around each other, in the form of helix. It looks like staircase for us, helical staircase. So DNA structure, two strands, they are helically coiled. We call them as double helix structure. Then you have small units in the DNA structure. They are called as nucleotides. Each unit of nucleotides or each set of nucleotides has 3 subunits in it. It has sugars, it has phosphates and nitrogen bases. Then this is what is one unit of nucleotide. You have base, nitrogenous bases, sugars. The sugar is pentose sugar. If you see, you have 5 sides. So this is pentose sugar, or I can also call them as deoxyribose sugar. And your phosphate groups. Phosphate groups, sugar and your nitrogen bases makes up one nucleotide. Continuing with DNA structure. The DNA structure consists of 4 nitrogenous bases. As I said, nucleotide has a sugar molecule attached with base and on the other side phosphate. So those 4 bases are nitrogenous bases. They are adenine, guanine, thymine and cytosine. You might be noticing I have written adenine with one color and thymine with one color, which means adenine forms 2 hydrogen bonds between themselves. That is, adenine forms 2 hydrogen bond with thymine. Then cytosine forms 3 hydrogen bonds with guanine. That's why I have written both with the same color. So this 4 nitrogen bases forms base pair among themselves. As you are seeing this in the DNA structure. The ladder-like structures are nothing but the base pairs of this nitrogen bases. If you see adenine, base pairs with thymine. This is next alternative. Everything will be alternative. Here adenine is base pairing with thymine. Then the alternate one, thymine is base pairing with adenine. And in the second one, if you see, cytosine forms triple bond with guanine. And then in the next alternate one, guanine is forming base pair with cytosine. So everything will be the alternate one, the opposite ones. In the next ladder, if you see adenine, thymine again, cytosine, guanine, thymine, adenine, guanine, cytosine. The structure of DNA, as I already said, this is one strand, this is one strand, this is one strand. Okay, these are the nitrogenous bases. The space between one base pair and the second base pair is 0.34 micrometers. Then the total, this is your 2 units of DNA. This 2 units of DNA which has 5 base pairs totally in each, that is totally 10 base pairs, are 34 micrometers totally. Coming back to this, new strand is formed by replication. See, this DNA has an ability, or the common nature of this DNA is to replicate and form new DNA. So how is it formed? Replication of DNA, we have already seen during interphase, the double helix, one end opens at one end and two strands becomes free. See, if you see, let me take this end. This end, if you see, it is coiled around each other. Now during interphase, this coil uncoils, and this end becomes free, this end becomes free. If you see, this end becomes free. When we are freeing this, the above one will also get freed up continuously. First this will get freed, this will get, next this one. So it comes in a sequence. So this becomes an old strand, this is one strand, and this one will become the second opened strand. So during interphase, the helix opens at one end and two strands becomes free first. Then new nucleotide starts forming complementary strands. Complement the additional. So to the old strand, if new strand is being formed, assume the old strand is from 5' to 3'. The new complementary strand will start from 3' to 5'. The opposite, the new one that is being added is your complementary strand. Complementary strands around old strand is called as DNA replication. DNA replication is the main part of this DNA structure. That's the end of this chapter. Chapter 2, cell division, cell cycle and structure of chromosomes. In my next video, I will be telling few important questions and weightage of this chapter for your board exam. If you like my videos, please click like button and share it to your friends. Don't forget to subscribe my channel. Thank you for supporting me.